Assalamualaikum, this is Nan and today we're going to talk about insulin. We're going to talk about the synthesis of insulin. We're going to talk about the working of insulin. We're going to talk about the effects of insulin on different types of metabolism, on growth. We're going to talk about the secretion of insulin. And lastly, we're going to talk about the regulation of insulin along with some fun facts. So let's start. Insulin. Insulin is a protein having a molecular weight of 5808, consists of 2 amino acid chains and have a half-life of 6 minutes in the plasma. Now, insulin is synthesized in the beta cells of the pancreas. The precursor, that is proinsulin, consists of leader chain, A chain, C chain and B chain. When the leader chain is cut off by enzymatic activity, the proinsulin converts into proinsulin, which consists of A chain, C for connecting chain and B chain. Now further enzymatic activities in the Golgi apparatus causes the release of C chain and the formation of disulfide bridges between the A and B chain. This is the normal insulin. This which is packed in granules inside the beta cells and released upon subsequent stimulation. Now what happens to the C chain? The C chain has no insulin activity and binds to the membrane receptors like sodium potassium ATPase, endothelium nitric oxide synthase, and have other functions. Now working of the insulin. The insulin binds with the insulin receptor, which has 4 subunits, 2 alpha and 2 beta. The alpha one are outside while the beta one traverses the cell membrane and goes inside the membrane. Now the attachment of the insulin causes the phosphorylation of the subunits, which causes the activation of tyrosine kinase. Now the tyrosine kinase activates several insulin receptor substrate enzyme and along with it activates other enzymes. All these activities results in the increased glucose uptake, increase in amino acid permeability, increase in enzyme activity, increased translation and transcription of protein, all of which results in the increased blood glucose level. Here's the insulin receptor, which consists of 2 alpha units outside the membrane and 2 beta unit which traverses the membrane. When insulin attaches, it causes the activation of tyrosine kinase, which activates several insulin receptor substrate and causes the phosphorylation of enzyme, releasing in increased glucose transport, protein synthesis, fat synthesis, glycogen synthesis, growth and gene expression through the increased glucose influx and subsequently increase the blood glucose level. Now talking about the effects of insulin on carbohydrate metabolism. Now generally speaking, insulin increases the glucose utilization. It decreases the glycogen breakdown of glycogen by decreasing the activity of phosphorylase, which breaks down glycogen into glucose. So it ultimately decreases the glycogen breakdown. It enhances the uptake of glucose by enhancing the diffusion, and it traps the glucose inside the cell by increasing the activity of glucose kinase. Now this glucokinase adds a phosphate group to the glucose, and the phosphorylated glucose can't leave the cell membrane. Now it increases the glycogen synthesis by increasing the activity of glycogen synthase. Similarly, it decreases the gluconeogenesis because insulin has to decrease the blood glucose level, so it decreases the de novo synthesis of glucose by decreasing the gluconeogenesis. How does it do so? It decreases the quantity and activity of the liver enzymes which do the gluconeogenesis, along with the decreased amino acid release due to insulin, since amino acid act as a substrate for the gluconeogenesis. So It also converts the excess liver glucose to fatty acids, which are then transported via the lipoproteins to the adipose tissue for storage, since insulin is an enzyme of well-fed state. Now, moving toward the fat metabolism, insulin is called the fat sparer because it utilizes glucose for energy in place of fat. So it increases the fat synthesis by increasing the glucose transport inside the cell. This glucose transport causes the utilization of glucose in glycolysis, which produces the substance called the pyruvate. Now this pyruvate undergoes subsequent conversion to acetyl CoA. So it increases the amount of acetyl CoA, which act as a substrate for the fatty acid synthesis. Now, fat metabolism, the insulin also increases the triacyl glyceride synthesis because glucose provide the glycerol portion which is required for tag synthesis. Since it increases the synthesis, it should decrease the hydrolysis of tags. So it decreases the hydrolysis of tags. How it do so? It does so by decreasing the activity of hormone sensitive lipase enzyme, which decreases tags into fats and glycerols. So now moving toward the effect on the protein metabolism, the insulin increases the amino acid influx inside the cell. So it provides a substrate for protein metabolism. It increases the translation and transcription. It decreases the protein metabolism, conserving the protein components of the body. And as I have explained earlier, it decreases the gluconeogenesis so that it can maintain the blood glucose level. Now, the insulin has also an effect on the growth of a person. Insulin works synergistically with the growth hormone to increase the growth via the protein formation, as I've explained earlier, by increased amino acid influx. Now talking about the secretion of insulin. Insulin increases when the glucose concentration increases in the blood, which is a prime controller of insulin secretion. The glucose moves inside the cell via the glucose transporter 2. So glucose influx occurs. Now this glucose is acted upon by an enzyme called glucokinase, which converts it into glucose 6-phosphate, and this is the rate limiting step. The glucose 6-phosphate is oxidized via a series of reactions to causes an increase in the adenosine triphosphate concentration. Now this adenosine triphosphate acts upon by the adenosine triphosphate sensitive potassium channel, which closes it, and the closure of potassium causes the depolarization of the membrane. Now this depolarization opens the voltage sensitive calcium channel. When this opens, it causes the increased influx of the calcium, which fuses with the insulin containing vesicles and causes the exocytosis of the insulin. Now an important point to note here, that is the drug sulfonylurea also blocks the ATP sensitive potassium channel, so it increases the insulin secretion. Again, increased glucose concentration causes the increased influx through the glucose transporter. The glucose is phosphorylated, then oxidized to cause increased concentration of ATP, which blocks the ATP sensitive potassium channel, and this causes depolarization, which opens the calcium channel, and this calcium fuses with the vesicles containing insulin to causes the exocytosis of the insulin. Now regulation of insulin. Regulation includes the stimulatory factor and inhibitory factor. Now as we have explained earlier, insulin causes the increased utilization of amino acid. So increased in the amino acid in the blood will cause the secretion of insulin to cause its increased influx. Similarly, the prime controller of the insulin is the increased blood glucose level, and since insulin also causes the fatty triacyl glyceride synthesis, for which we need fatty acids. so free fatty acids increased concentration in the plasma causes the secretion of insulin. Now another important factor is the gastrointestinal hormones and secretin, gastrin, whose concentration causes a positive impact on the insulin. These causes positive impact on the insulin because as an anticipatory increase, so that when we eat and our blood glucose level rises, there should be enough insulin to control the blood glucose level. Now beta adrenergic stimulation increases the insulin working via the, since these are the G proteins, class of G protein, so they activate a cascade of enzyme reaction which increases the insulin action. Now parasympathetic stimulation, which produces the acetylcholine, increases the insulin stimulation. Similarly, hormones like growth hormone and cortisol, which are produced in response to hypoglycemia just like insulin, increases its activity. Now another important point here is that glucagon increases the insulin secretion. It increases the insulin secretion indirectly because it increases the blood glucose level, and increased blood glucose level causes the increased secretion of insulin. Now moving towards the inhibitory factors of the insulin, the decreased blood glucose level causes the decrease in the insulin production via negative feedback. During fasting, the blood glucose level is low, so the insulin production will be halt. Now alpha adrenergic receptor is a class of G protein coupled receptors which activate a cascade of protein enzyme reactions which ultimately decreases the insulin secretion. Another important thing here is that the somatostatin, which is produced by the delta cells of the pancreas, it decreases the insulin production. Actually, the somatostatin is used to control the secretions produced by the insulin. Moving toward the fun fact portion of our topic, a question here is that in giant people, in giants like acromegalic, why diabetes mellitus develops? So the answer is in response, in giants, the growth hormone is excessively secreted, and as we know that growth hormone causes the increased secretion of insulin. This secretion of insulin is so much that the beta cells of the pancreas undergo exhaustion, and the exhaustion of beta cells causes the decrease in the required amount of insulin in the blood. So the body cannot regulate, the body becomes unable to regulate the blood sugar, and this ultimately results in diabetes mellitus. The similar effect is also included in people with high doses of pharmacological drugs, and also in people with the excessive release of cortisol. And that's all for today's lecture. Please subscribe the channel, and thank you. Yep.